Hi, my name is Ian Miller. I'm a pediatric epileptologist who is working for Marinus, and it's my pleasure today to share with you information regarding Ganaxolone, which is an investigational drug for CDKL5 deficiency disorder. We are going to review today the Ganaxolone molecule itself. We are going to review clinical evidence for its effectiveness, specifically the Marigold study. We're going to look at summary information, and we're going to take questions, which I believe will be offline. So to start, I just want to familiarize you with the Ganaxolone molecule itself. It is a synthetic neuroactive steroid, which is in clinical development for both acute and chronic seizure disorders. It has been in development for quite some time for a number of indications, and we are excited because with all of that information that's been collected, we have a lot of information about its safety and tolerability. In addition, CDKL5 deficiency is the indication for which we have data that will allow us to go for approval. And so that's the data that we're going to review today and the mechanism by which it can help CDKL5. So to that point, I think most, if not all of the audience members today have a loved one or maybe even a family member who's affected by epilepsy. We might not think very deeply, however, about the underpinning mechanisms behind the epilepsy. And in general, I'm going to kind of cover this slide in a high level, and then we're going to look at some figures and diagrams on the next slide. But epilepsy is a disease which happens when the excitatory systems in the brain are overactive or the inhibitory systems of the brain are underactive. So there are two strategies that you can employ in order to stop seizures, either increasing inhibition, which turns down the brain cell activity, or reducing activation, which will also reduce brain cell activity. And Ganaxolone works through GABA, which is the primary inhibitory neurotransmitter in the brain. So what does that mean? Let's take a look at a picture. So GABA, which is abbreviated GABA here on the middle of the top left diagram, is the primary inhibitory neurotransmitter in the brain. And we have lots of medications on the market that work through a GABAergic mechanism. And we therefore know with a lot of certainty that medications that affect these receptors can be very effective anti-seizure medications. The currently available medications are things like clobazam, diazepam, lorazepam, midazolam. The whole benzodiazepine family work on GABA, as does phenobarbital. So one question is, why would Ganaxolone be beneficial if there's lots of GABAergic medications that are on the market today? And the reason is because Ganaxolone not only works on the blue GABA receptors, which are the synaptic receptors, which are shown here, and synaptic simply means they're in the synapse between the cell that's relaying the message and the cell that's receiving the message, but also the extrasynaptic, meaning outside the synapse, which are shown here in green. So that's really what's unique about Ganaxolone. It is a GABA antagonist, but it works not only in the intrasynaptic receptors, but the extrasynaptic receptors. And that is the hypothesis behind bringing it to clinical availability and clinical use. And we'll review in the next several slides the data that supports the idea that this is an effective anti-seizure medication. So to that point, the data was collected in the Ganaxolone study in CDKL5 deficiency disorder, which is called Marigold. It was a global phase 3, double-blind, placebo-controlled clinical trial that evaluated the use of oral Ganaxolone in children and young adults with CDKL5 deficiency disorder. A total of 101 patients were enrolled between the ages of 2 and 19 years. All had confirmed disease-related CDKL5 mutations, which caused their epilepsy. They had a baseline number of seizures that was 16 or more major motor seizures per month, and they were taking up to 4 concomitant anti-seizure medications. So these individuals were uncontrolled on all of the conventionally available seizure medications. And when you do a scientific study, you need to declare ahead of time what your primary endpoint will be. And that endpoint was chosen to be the % change in 28-day major motor seizure frequency. So what happened? Well, before we get to what happened, let's review an overview of kind of the study design and the timing of it all. So we already said that patients had to have 16 seizures per month for 2 months prior to the study, after which they started a baseline period where they actually kept track of their seizures for that 6 weeks. And if they had a sufficient number of seizures, they were eligible to be randomized. The diagram only shows kind of one line, but really there's 2, one for the placebo group and one for the treatment group. The assignment was randomly allocated for half and half, and the individuals who were on placebo were titrated up to placebo, and the individuals on ganaxolone were titrated up on ganaxolone, after which for 13 weeks, that dose was kept the same of whatever product they were on. At the conclusion of the maintenance period, they were titrated down and were eligible to continue into the open label phase, where all of the subjects received medication. The total of the maintenance phase plus the titration phase is the treatment phase of the double-blind portion of the study. And most of the data that we're going to review takes place in this double-blind phase. However, we will look at some open label data, which gives us some sense of the long-term effectiveness and how it changes over time. So this is the first outcome slide, which is the most important one. This is the primary outcome measurement, which again, we said was the % reduction in major motor seizures in a 4-week period. So this shows that patients on ganaxolone, shown in teal, had a 30.7% reduction in major motor seizures, and it shows that the placebo group only had a 6.9% reduction in major motor seizures. So that difference is statistically measurable and clinically significant, and so that is the reason that we're so excited about the availability of a new medication, which is specifically demonstrated to be effective in CDKL5 deficiency. So if we look at other outcomes for the study, a secondary endpoint was the seizure severity. And this was simply a caregiver global impression of change in seizure intensity and duration. And you can see again that the teal group, which is the treatment group with ganaxolone, and the gray group, which is the placebo group, does separate and behave differently. If you received the teal, the medication, then you're in the teal group, and you're more likely to be on the left side of the curve where there is improvement, either very much improvement, or minimally improved, versus no change, which is here in the middle. Most of the placebo patients were in the no change group, and it's kind of a bell curve with a few that got better, a lot that didn't change at all, and a few that got a little bit worse. And so again, we can hand this bar chart over to our statisticians, and they're able to tell us that statistically there is a measurable difference, which is meaningful. So again, that was a positive result, which shows that CDKL5 seizures can be effectively treated with ganaxolone using this outcome score as a way to document that. So I think the last efficacy slide with the double-blind phase that I'm going to share with you is this one. This simply looks at different subgroups in the Marigold study. The first is subgroups based on region, and you can see that the teal bars are essentially the same in all 3 regions that were studied or broken down: the US, Europe, and Russia and Poland. You can see that the subgroups from male versus female were not significantly different. And you can see that the subgroups based on how many seizures they had at baseline are not notably different. And here the groups are less than 35 seizures, 35 to 93 seizures, or more than 93 seizures in a 28-day period. And then this is the final outcome slide and effectiveness slide that we're going to look at. I told you that most of the data would be in the double-blind phase. We were finished with that. We're now shifting gears and looking at the open label extension phase. And the reason that that's important is because we have a chance to see, number 1, how long the treatment effect persists. Number 2, how the placebo treatment group responds once they're put on active medication. And number 3, we're also able to collect more information about safety and tolerability. So ganaxolone demonstrated durable seizure reduction in the open label extension, which is best shown in this plot. The original data that we already talked about was 30.7% versus 6.9%, which is the first 2 bars. And as you look after 1 and 2, 3 and 4, 5 and 6, 7 and 8 months, etc., you can see how those curves change. And what I'm happy to see as a doctor who takes care of these children is that that bar does not shorten over time. If anything, it gets a little bit more robust. The other thing that's very heartening to see is that the patients that were on placebo and did not have a very big reduction in their seizures started to have an improvement in their seizure control because they started the open label treatment. So that also suggests that the ganaxolone is effective, consistent with the double-blind data. So patients treated with ganaxolone for at least 12 months experienced a median 49.6% reduction in major motor seizure frequency. Patients transitioning from placebo to ganaxolone demonstrated seizure frequency improvements, and there were no new safety findings that emerged in the open label extension to date, which is also exciting, even though it's exciting in the tolerability part of the medication profile and not the effectiveness part. So let's talk more about the safety findings that we're seeing primarily in the double-blind phase, and those are summarized here. On the left, you can see treatment emergent adverse events, and on the right, you can see serious treatment emergent adverse events. Keep in mind that treatment emergent adverse events is anything untoward that happened during the double-blind phase. And so that happened in the placebo group, and it happened in the ganaxolone group, and we're never certain what is responsible for adverse events that we see, because it might be the underlying disease, it might be the investigational product, or it might be neither. It might simply be a coincidence. So you can see in ganaxolone and placebo, any treatment emergent adverse event happened in 88% of patients on placebo and 86% of patients on ganaxolone. So that's already reassuring, because the overall number of adverse events are not notably higher in the ganaxolone group. There was a suggestion of somnolence in the treatment group, with 36% of patients on ganaxolone and 15.7% of patients on placebo with that adverse event. And pyrexia and upper respiratory tract infection also had a higher percentage in the ganaxolone group than the placebo group. So those are all treatment emergent AEs. If we restrict our focus to serious treatment emergent AEs, we get a smaller number of adverse events that are seen, because most of them are not serious. And we can look at the aggregate numbers overall, both in placebo and ganaxolone. The placebo percentage is 9.8, and the ganaxolone percentage is 12%. And the breakdown is as shown. Because we've kind of filtered out the less severe adverse events and we're only looking at the serious ones, it's much more difficult to look at differences between the two groups, just because each individual type of AE happens at a low rate. And so you have kind of a 1 or 0 in most of the places where it happened once or not at all. And that's very hard to compare and draw conclusions about what's responsible. But the list is shown there, and you can kind of draw your own conclusions about, kind of what that means and how it compares to other seizure medications. In my clinical judgment, it's a very favorable adverse event profile. So in summary, ganaxolone is an investigational drug that's in development for the treatment of CDKL5 deficiency disorder and other rare seizure disorders. In a randomized, double-blind clinical trial of CDKL5 deficiency, patients taking ganaxolone had significantly fewer major motor seizures than patients taking placebo. Ganaxolone was generally well tolerated. Patients in the ganaxolone group experienced a discontinuation rate that was under 5%, with somnolence being the most common adverse event. Ganaxolone continued to reduce seizure frequency over a 12-month open-label extension of the study. In the United States, Marinus is sponsoring an expanded access program for eligible patients with CDD to receive ganaxolone under a treatment IND. And Marinus plans to submit a new drug application to the FDA mid-year for ganaxolone in CDKL5 deficiency disorder, and intends to file in Europe thereafter. So with that, I thank you for your attention, and I'd be happy to take any questions that you have through the mechanism that's been provided. Thank you.
Protein, the biotin ligase, BRA, will add the biotin molecule to those proteins. So we can take that the total set of proteins, digest them, and then pull down specifically the proteins that have been biotinylated. We can then use tandem mass spec analysis to sequence those peptides, and map them onto the actual proteome of the cell. And from there, we can suggest which proteins have interacted closely with CDKL5. We've tested several different configurations of the biotin ligase, either on the N terminus or the C terminus of CDKL5, with and without the TAT cell penetrating peptide. And although I'm not ready to share the entire data set, there's still a lot of work to do. We've really only just started to scratch the surface of the protein-protein interaction analysis that we can do. There are early indications that the CDKL5 has an interesting connection to mitochondrial targets. And then one other note would be that the two centrosomal proteins, PCM1 and CEP131, are the only genes that were enriched in all the test configurations, no matter which side of CDKL5 we put the biotin ligase, or with or without the TAT cell penetrating peptide. Those two genes always showed up. Our future plans include target validation, in vivo interactome analysis. So we plan to deliver a CDKL5 BRA fusion protein to a mouse and do a very similar experiment, as well as some other proteomic tools. Now, in the last slide, I'll talk about our CDKL5 protein therapy. As I have presented in previous conferences, we spent a long time, 4 or 5 years, working on the significant challenges to express and purify milligram quantities of full-length active CDKL5. We had success when we moved to the baculoviral insect cell system and increased our expression by orders of magnitude, and simplified and focused our purification method to reduce the number of steps required to get to a greater than 90% purity. So once we did that, we spent the last year to year and a half working on developing assays, such as an in vitro kinase assay, a mass spec-based detection assay, and as I've shown already, using it to evaluate cell uptake and attempting to look at phenotypic rescue of cells in culture. But now we're at a point where we're ready to think about it as an ERT candidate more and design experiments to assess its pharmacology and look at biochemical or behavioral rescue in animal models. And currently, we're in the early stages of planning those mouse studies with purified TAT CDKL5 as ERT. And I am very much looking forward to sharing more data as it becomes available. To conclude, I'd like to thank what you can see is a long list of people at Amicus, at Penn, and at Sanford Research who we collaborated with to run the BioID experiments. In fact, that lab invented BioID. Without the help of all of these people, and I'm sure I'm forgetting some, we would not be where we are today with this program. We have a lot of work left ahead, but I'm extremely excited about the prospects and very much looking forward to the next year of drug development. I am happy to field any of your questions, and I'd like to thank you for your attention today. Take care.